Hello everyone, my name is J.Flynn. I am a graduate student in the lab of Jason Brenchley at the NIH, and today I'm going to be talking about my work on the isolation and proteomic profiling of translocating bacteria in progressive SIV infection of Rhesus macaques. Before diving into my own work, I wanted to provide some background on microbial translocation first. So microbial translocation is the movement of bacterial metabolites and whole bacteria from the gut lumen into systemic circulation after passing through a compromised GI tract, such as what occurs during SIV and HIV pathogenesis. Factors behind this include the loss of tight junctions and enterocyte death, which leads to this kind of characteristic feature of a leaky gut. And so once these gut-derived bacteria are able to spread into systemic circulation, they can lead to peripheral immune activation, which may be an underlying cause of some of the HIV and SIV disease progression that we see, as well as poor immunological responses to antiretroviral therapies, and may contribute to some of the non-infectious comorbidities we see, such as neurological impairment and cardiovascular, liver, and bone diseases. Previous work done in my lab showed that microbial translocation did not appear to be a random process. So in this early study, they tested for 16S rDNA signatures in the gut lumen versus the lamina propria, which would show those species that were translocating. And what they found was that what was able to escape into the lamina propria was not simply representative of what was in the gut lumen. In particular, you see these much higher abundances of the proteobacteria in both of the animals pictured here in the figure. Then in some of my own work, I've been able to isolate and identify 36 species of bacteria that have escaped from the gut and entered the systemic tissues in liver, spleen, and mesenteric lymph nodes of SIV-infected Rhesus macaques. And so from this previous work and some of my own, we got to wondering, why does this process not seem to be random? Is there something that allows certain bacteria, such as the ones I've been able to isolate, to preferentially translocate? And this is the question that we really wanted to get at in some of my ongoing studies. And to get at this, we decided to take a metaproteomics approach. So we sent 8 isolates that were translocating species, pictured here in red, and 5 non-translocating species, pictured here in blue. And these were the non-translocating species were simply isolated from the stool of animals, and these were selected for species that had not been shown to be translocating. So they hadn't come up in any of the 20 Rhesus macaques that we've analyzed. And we took cell pellets from these and sent them to the Bergener lab for proteomic profiling. So after the proteomic results came back, it was determined that there was a unique bacterial protein expression signatures between the translocating and the non-translocating species, with 5,103 unique proteins detected across all the samples included in the study, and approximately 47% of those, 2,400 of them, being identified in translocating bacteria and being unique to those bacteria. And when their bioinformatics team did a closer look at these results, we determined one of the top 2 hits was a cytosine-specific methyltransferase. The other was a copper homeostasis protein, but we decided to focus on the cytosine-specific methyltransferase as a potential novel mode of translocation. And we decided to focus on this because of the role of methyltransferases in epigenetic modifications. And so from previous various studies, we know that epigenetic modifications of gut mucosal cells can lead to large-scale changes, which may allow escape of the bacteria and their different metabolites, such as contributing to loss of tight junctions or enterocyte death, or the bacterial methyltransferases could be causing modifications within the bacteria themselves or to surrounding bacteria, depending on if the methyltransferases are secreted or simply held within the cells. And if they are doing internal modifications, it could be for things such as altering the expression of flagella genes or altering their metabolism to better survive in the host body versus the gut lumen, since these are very different environments. As a next step to these findings, we wanted to determine how inhibition of these methyltransferases may affect the growth of some of the translocating species that I've been able to isolate. And here I've pictured a few representative species. On one end of the spectrum, with the Streptococcus luteatiensis, you can see that they really didn't have much of a response to inhibition of the cytosine-specific methyltransferases. rather resistant to any inhibitory effects. Whereas when you move to the Citrobacter frundi, which had been shown in the metaproteomic data to have some of these cytosine-specific methyltransferases, it has this really nice dose response with 5 millimolar having quite a significant inhibitory effect, and it really only recovers regular growth once you get down to about the 50 micromolar concentration of the inhibitory molecule. And then really on the other end of the spectrum from the Streptococcus lutetiensis, you have this isolate Weissella confusa, which was not able to grow in the presence of the inhibitory molecule until you get down to 500 nanomolar concentration, and even then, its growth is nowhere near what it's able to do in the normal media. And so from these, we draw a few main conclusions. So protein expression between the translocators and non-translocators is distinct. Inhibition of cytosine-specific methyltransferases can have significant effects on the in vitro growth of translocating species. And bacterial-derived methyltransferases may represent a novel mode of microbial translocation, and we're hoping to provide better evidence for this in future studies. And with that, I would like to acknowledge members of my lab, especially Jason Brenchley and Alex Ortiz, for all of their mentorship and guidance through the project, as well as the NIAID Microbiome Program, the NIAID BCBB, all of our veterinary staff who help us with samples, and of course the Bergener Lab for doing all the metaproteomics work and analysis for us. And with that, thank you for this opportunity to present my work, and I am happy to take any questions during the Q&A to follow. Thank you very much.